Dear students, good morning to one and all. Today we are going to learn about Human Genome Project. This is Dr. S. Pravina, Department of Zoology, St.Joseph College of Arts and Science, Autonomous, Cuddalore. So first we are going to learn genome. A genomic means is nothing but is the studies of mapping, a sequences and analysis of the genome. So the chromosomes have contains many genes that we know very well. It is the scientific disciplines of genomes of cells studied through the high thought sequences. It is help to understanding the structure, function, and coordinated regulation of all the genes is an organisms. So a genomic is the entire gene of an organisms. Genes carry informations for making all the proteins required by the organisms. This protein determine among other things how the organisms look, how will its body, metabolisms, food, fights, infections, and even how to behaves. The DNA is made up of a four similar chemical. We know that and all. It's adenine, thymine, guanine, and the cytosins that are repeated millions or billions of times throughout the genome. The human genome, for examples, has 6 billion pairs of the genome. The particular sequences of adenine, thymine, guanine, and the cytosins it's extremely important. These sequences bring out life diversity, differentiations of one species from other, evolution of the organisms, local variation of the species. So, introductions. Human Genome Project was internationally scientific research project which got successfully completed in the year 2006 by sequences the entire human genome of 3.3 billion base pair. The Human Genome Project led to the growth of a bioinformatics, which is a vast field of research. The successfully sequences of the human genome could solve a mystery of many disorders in the human being and gave us the way of go up them. So Human Genome Project it's involved the discovery and the sequences of full DNA completed in the single human somatic cells. It is primary goal listing the locating of all our genes in genome of any given individuals except the identical twins is unique. Mapping the human genome involving the sequences multiple variations of the human genes. So what are the main goal of human genome projects? First one, goal of the human genome projects including optimized of the data's analysis, sequences the entire genomes, identification of the complete human genomes, creating genome sequences databases to store the data, taking care of the lethal, ethical, and social issues that the project may pose. So, the project was planned in the year 1986 by the Charles Dilsey, who was the director of the US Department of Energy DOE. The DOE National Institutions and Health Hand in the started in this project in the year 1990. The 1987's report said broadly the ultimate goal of this initiations is to understand the human genomes and knowledge of the human genome necessary for the progressive medicines and other health science as knowledge of human anatomy. At least 18 countries have participated in the human genome research project. They are Australia, Brazil, Canada, China, and so on. In fact, major part of the research has been conducted in the different laboratories in these countries. Some developing countries have participations through the studies of molecular biology, techniques for genome research and studies of the organisms. The Human Genome Project organizations help to coordinate the international collaborations of human genome projects. So the complete set of the genes, what are the process doing this project? So first one, the complete gene set was isolated from the a particular particular organisms. It was then split into small fragmented. We are using the restriction endonuclease. That nucleus is easily cut a small bit of the fragments. This DNA structures was then amplified with the help of a vector which mostly was bacterial artificial chromosomes and yeast artificial chromosomes. And then smaller fragments were then sequences using DNA sequences on the basis of overlapping regions, and the sequences were they arranged. All the information of genome sequences were stored by computerized based on the programs. This way the entire genome was sequences and stored as a genome database in this computer. Its a genomic mapping was the next goal which was achieved with the help of the microsatellite. So, what are the futures in the human genome projects? So, our entire genomes is made up of 3,164.7 million base pairs. An average gene made up to 3,000 nucleotides. The functions of more than 50% of genes is yet to be discovered. A protein code or non-protein code also identified. Most of the genome is made up of repetitive sequences which have no coding purposes specifically, but such redundant codes can help us better understand the genetic development of the human throughout the genes. A chromosome 1 has most number of the genes and the chromosome Y has a least through the 231 genes only having a human male chromosomes. The chromosomes 1 having a multiple genes, it contains a 2968 genes, it's having a first chromosomes. A human genome project has identified 1.4 million locations with a single basis DNA differences in humans. These informations will revolutions and identifications of many diseases in the tracking of human histories. So we have using two different methods for in the human genome project. Then first one is the Sjctnc method. So DNA synthesis is carried out the presence of the limited amount of fluorescence labeled ditetraoxyribonucleoside triphosphates that results in chain terminations through the chain terminations fragments of distinct ties and generated via small separated by gel electrophoresis. So both so Sankar math sequences is also known as a chain terminational method. Here DNA replications is stopped at positions occupied by one of the four bases of the resulting fragment length are analyzed. This process involved using an enzymatic procedures to synthesize a DNA chains of variance length of four different reactions. So next we are going to learn a Maxim Gilbert chemical degradation method. So Maxim Gilbert and Sankar techniques are first generation sequences techniques. So Sankar by chemical degradations is known as chemical sequences. It was published in the year 1977 by Alan Maxim and Walter Guilford. It's requiring chemical modifications of the DNA and further cleavage and the electrophoresis. The sequences of polymerase gel electrophoresis and autoradiographic allows the assess a single stranded DNA fragments separated by the size of the directions and radio labeled and DNA band pattern of an X-rays film and encoding the DNA sequences. Identify this kind of methods. Thank you.